Hi, I am Mridula Jayaraman, and I go by MJ. I am a developer, and I work for ThoughtWorks, who is a software consulting company. I'll tell you a little more about myself and my company later on in the presentation. But I'm here today to talk to you about a project I got to work on over the last year and a half. And in this way, I learned that developers can also help treat ovarian cancer. So it was very exciting, and I hope you will be too. So an overview of what my talk's going to be about is I'll introduce some of the players involved to set some context. And in lieu of this, I'm also going to talk a little bit about some concepts in genomics and cancer that's relevant to the domain. And then introduce the problem we were faced with, our solution, some key learnings, and then questions. Cool. So the key players. Let me introduce to you the Clearity Foundation. They're a not-for-profit organization started by a woman named Laura Shaver. She's an ovarian cancer survivor and a scientist. And having faced the treatment and all its troubles and turbulences, she decided to come together. She joined along with other women scientists and started Clearity. So their goal is to help relapsed ovarian cancer patients find better alternate therapies for themselves. So their problem was that they came up with a good model to work with the constantly growing flux of next generation sequencing data and other findings in the cancer realm. But their trouble was their lack of technical competence. So they were able to treat about 10 women a month, but had the potential to scale this up, and their goal was to do 1,000 women a month, but they didn't have the expertise to help automate this and take what was in their brains and actually put it into a computer algorithm or sorts. So they met with another company called Untitled Systems. They're a startup based in the Bay Area. And what they really wanted to do is, again, to help oncologists and other researchers who are working in the cancer realm come up with ways in which they can commercialize and actually go help patients with cancer and help them, again, come up with better alternate therapies for these patients. This still doesn't introduce me or ThoughtWorks, and I'll get to that. But before that, we talked about cancer. We talked about there's a huge word genomic here. So I'm going to jump a little into the concepts of genomics and cancer that's relevant. So the first thing is DNA. So in 2003, the first human genome was sequenced. It was a very expensive process, but it happened, and that was very exciting. So what is a genome? A genome is essentially comprised of DNA. DNA is sort of the building block of every single human being. And for our purposes, let's think about it as a continuous stream of characters. And there's 4 options, A, C, G, or T. It's okay if you don't remember any of this, but just think of it as continuous lines of A, C, G, or T going over and over again. So we've spent a lot of time and effort understanding this. Why is this important? DNA is what codes for the functional part, which is a protein, and different proteins come together and make us who we are, and make us function and define us, essentially. So portions of the DNA, oftentimes we refer to them as genes. And this categorization is essentially, oftentimes when we think gene, it's that part of the DNA that ends up coding for a functional protein. So a functional protein is something that is then involved in some other process to help us function as human beings, plants as plants, etc. So in this process, the base DNA that today we're getting better and better at understanding goes through many processes before it can become a protein. And very quickly, I'm going to introduce this process. So a DNA, essentially, imagine that it gets cut up and relevant parts are put together to first form what is called a pre-mRNA. So it's essentially the same strand of DNA becomes mRNA. This process is known as transcription. So there's then post-processing, where some other irrelevant parts are taken out, and then you put together just sort of the coding sequence, which eventually can be translated into the protein. Again, it's okay if the exact details is not very clear, but the important thing that I want to point out here is that we today are understanding this part really well, but there's still a lot that happens before we get to a protein. And this is just the happy path. In life, actually what happens is this process of alternative splicing. So based on which cell it is and what type of process is involved, your gene or your DNA is cut up in many different combinations and can result in many different proteins. So I want to introduce another terminology here. Oftentimes this part up here, so whatever is your particular genetic sequence at a DNA level, is referred to as your genotype. And then what ends up coming out as a protein is referred to as your phenotype. So for example, you have a gene that codes for eye color, and a lot of us have heard about this example where you have a dominant allele and a recessive allele. Again, I don't want to get into too many details, but your genotype can have the dominant and the recessive, so two options. And your phenotype is one of the two options actually showing up and you having a particular eye color. Cool. Why do we care? Because, again, we've gotten better at understanding DNA. One thing, again, I want to bring up is that it is a lot of data. So DNA, like I said, was a continuous sequence of ACGT. And actually, we have 3.2 billion base pairs in our 23 chromosomes. Humans actually have 46, so 23 times 2. So we have double this amount of base pairs all in all. But we believe we can be defined by just 1/2 of it, assuming they're exactly the same. And this is all for normal, ideal, happy path scenario. So out of these 3.2 billion base pairs, 3% is what actually ends up as proteins or functional proteins. So in effect, today, we're mainly interested in about 25,000 genes that actually that we believe code for proteins that end up affecting our bodily functions. And finally, as I had mentioned, we're getting better at sequencing. But today, this data amounts to about 1/2 terabyte for one genome. So when you sequence, you don't just read things once. They read it over and over and over again. So altogether, this ACGT, you have about 1/2 terabyte of this data. Cool, easy, huh? So great. Again, why are we learning about DNA? Because of mutations and their effect on us. So we have these continuous lines of sequences, but each time your cell divides, or in general, based on other environmental factors, changes happen. Evolution has caused changes. In general, every single day, each of us are probably undergoing some form of mutations in our cellular level. A mutation is simply a change at a base pair. There's more high-level definitions of this, but I'm giving you the simplistic one. So assume in a particular gene, at one particular position, something changes. You can call that a mutation. We have normal mutations. So like I said, every day our cells are dividing and things are changing. Mutations can happen. And we have about, I would say, I think it's 50,000 normal mutations on average. And that's normal. So between myself and someone sitting here, there's a lot of differences. And those are normal differences that we oftentimes call germline mutations. But the place where it gets dangerous is when we get cancer. So cancer is a disease of abnormal gene function. So it is when modifications happen in your DNA, which ends up affecting cell growth or cell death, we get cancer. So our cells start dividing out of control. And as they divide out of control, they're causing more and more changes. So compared to your normal genome, the cancer genome is quite different. There's lots of changes happening there. And there's also some important changes there that are actually causing a phenotypic implication that is resulting in this abnormal cell growth that your normal cells don't have. So I want to point out that today we've gotten better at understanding how to look at the genotype and be able to understand more about the type of cancer and its prognosis. But the place where there is still a huge, huge gap is in treatment choices. So before I get more in detail for that, I want to just talk about ovarian cancer. Like I said, the people I was involved with was Clearity, and they were an ovarian cancer treatment organization. Some facts about ovarian cancer is that it is a very lethal type of cancer. It's one of the 5 most common cancers affecting women today. In the US, per year, there's about 22,000 women who are diagnosed with ovarian cancer. The thing that's really dangerous about this is that symptoms are very hard to detect when this cancer is in early stages. So people get diagnosed. When they get diagnosed, they're already in advanced stages of this cancer. And out of this, about 14,000, and this number actually went up a little last year, will die. Actually, almost within 6 months of getting diagnosed. So it's terrible. The standard of care today is organ-based. So if they detect you have ovarian cancer, that is your abnormal cell growth started in the ovaries, treatment prescribed is targeted at your ovaries and less targeted towards the particular genotypic makeup that is causing this cancer. This is the case because, again, there are a few options. They have worked in the past. Research is still growing, but this is sort of what we best know today, and that's why it is like this. It's also incredibly expensive. So for ovarian cancer, there's 3 main chemotherapy options, especially when you're first diagnosed. Each of them costs about $100,000 per round, and they're actually prescribed pretty much by trial and error. So the oncologist will look at the patient, have some sort of feeling what will work, and then you go for the chemotherapy, and it takes a long time, and it's very painful. And there's surgery involved. Your lifestyle goes way, way down. Quality of life goes way, way down. And it might not work, and you'll just have to do it again. Also, this type of cancer will recur in about 80% of the women who get it once. So although your first round of treatment is successful, the chances of relapsing is pretty high. And the trickiest part is if you do relapse, your original options are not valid anymore because your cells are growing, and as they grow, they get resistant to these drugs you've already been exposed to. Okay, so this is very depressing, but things are changing. So research is growing. We're getting to understand details about how the cancer originated. We're also essentially growing so much in terms of how we're able to start sequencing and getting more information at a cellular level available even to oncologists. So I think today in the US you have about 600 next generation sequencing machinery available at the highest quality that we can come up with right now. The rest of the world combined is 500. So there's a lot of progress made in that angle, at least to get the data. And this has sort of been, in terms of expense, it's following the Moore's law. And we hope that within the next year, it's going to cost less than $1,000 to get any one of us sequenced and get that data available. So the gap here and the problem that we were faced with is, yes, you have the data. What are you going to do with the data? So it needs to be translated into a viable treatment option. Clarity and I then came to ThoughtWorks, because both of them had a good idea about how they can go from a mutation or a particular biomarker to a treatment, but they really didn't know how to scale this and commercialize this at all, and actually have production software, if you will. And this is when they involved ThoughtWorks and myself to come in and help provide the technical expertise to see this particular idea through. So it was very, very exciting, and we jumped right in, superbly excited to tackle this problem and hopefully get things going as quickly as we could. So when we started, I knew, this is the information me and my team had, is that we have a lot of data, and at bare minimum, we have about half a terabyte, which is the DNA sequence data. We do have access to some amount of cancer as well as normal sequence data for patients. And we knew from Clarity and from Anai some way of connecting different sort of aberrations in a patient's biology to viable treatment options. So we did have that knowledge. Great. So what did we do? We had to start building. Like we said, we could see how important it is, first the application itself, as well as this idea and this constantly changing field. We wanted to get something going as quickly as possible, given that we had even this slight amount of knowledge and this idea. So what we did, and I'm now going to actually start showing you what ended up becoming our end architecture, if you will. It's not really an architecture diagram, but something of that sort. And then I'll talk through the individual steps later. So first step was we wanted to parse all this immense amount of data. So we knew it wasn't enough if we just took what we had, which was the sequencing data. You had to contextualize it in order to do anything with it. So we tapped into many, many resources, parsed it. The thing that I quite enjoyed is we normalized this data. So we didn't choose one source over another. We grabbed them all. And then we try to unify them and take a union and try to have as much information as we could get. We then, again, which I quite liked, is made use of internal DSLs, or domain-specific language. I'll talk more about that later as well, to stuff it into our graph database. So great, we built a nice graph of all this information. And then we went about trying to deduce useful information from all this data we have. As we went along, we continuously kept a line of evidence. So in this world, it's not enough if you just say, here's an option. Contextualization was key. And we needed to enable the oncologist or whoever involved to be able to have a conversation with the patient and to also be able to cross-check the verification of any automated system, or the output of any automated system. So all this lived here. And so finally, when patient X could come along, we could give some treatment options. Cool. So step one was the data itself. So the language we chose to do the parsing of all, so this is, okay, sorry, I'm jumping points. But yeah, so the language we chose for parsing was JRuby. The reason we chose this is because we had a JRuby expert on our team. And so his name is Ola Bini. I think some of you might have heard him speak in other places. But we could definitely utilize his expertise to get parsing and sort of creation of these DSLs done very efficiently using JRuby. So the types of data we worked with was genes. So we went into all the public databases and we took all the information we could about genes. Most of them have downloads available. Some others we had to apply and get permission to take the information. We took information about genomes. We did biomarkers, which is any relevant biological indicator within a patient. So a biomarker can be something like a version of your gene. It can be a type of protein that's getting expressed very highly. So that entire sentence is sometimes called a biomarker. So it's kind of like a vague term, but we try to understand it better. We learned about drugs. We learned about treatments. Again, we went into patient data. We went beyond just DNA data. We took RNA sequencing data. We had test results available from Clearity for patients, as well as treatments they had already been taking. So the DNA and RNA data, again, we took publicly available data, as well as what Clearity patients had. And TCGA was our main source, which is the Cancer Genome Atlas. They're pretty amazing. They're releasing as many sequences as possible to help both research as well as some sort of treatment applications build off some sort of training model based off of this data. And then was the important data, which is treatment recommendations. So that connection between something in a patient and a viable treatment option. So we really had to tap into the brains of the scientists we were working with to actually get this into code. So this was fairly challenging. Honestly, to parse this data, it was us talking to them all the time, and then we put it into Excel files and then loaded it in. Cool. So then once you parsed it, I had mentioned we made use of internal DSLs. And I'm going to talk a little more using an example. So for example, consider the concept of a gene. So you'd read in genes from Ensemble. It's the name of a database. So you read many, many genes. So we parse all these different lines talking about a gene and then normalize the ones that are common into the same entity. So we basically parsed it and normalized it in Ruby, and we turned it into a data structure. So which was actually a Clojure DSL. So we outputted a line of Clojure code, essentially, which said gene that can have ID blah, name blah, properties XYZ. And this could then, when we decided to load it into our graph, Clojure would run. We'd expand that into an executable, it was a macro, so it would expand and it would load that data into our Neo4j database. So we would create a node named gene that can have a relationship of location to a chromosome, say 7, et cetera, et cetera. Cool. So like I mentioned, the choice for our graph was Neo4j. We knew this particular domain especially, it made total sense to look at things in terms of a graph. So you could again have a gene that has a DNA version, an RNA version, a protein version, a gene that's present in a patient, a gene that can have a particular mutation that can have a treatment option, and at any given point you can induce subsets of this graph to get information that's relevant to you. So along the next important step though is actually we loaded up the database, but now you needed to get the viable treatment option out. What did we do there? So in order to create what we called implications, the language we used is Clojure. And basically what we did here is link up everything in a meaningful fashion. And the most important, or sort of the magic here, was our ability to take any data point in a patient and be able to convert it into a patient biology marker that had a viable treatment option. Sorry, that's a really long sentence. So say someone has a DNA mutation of X. What we would do is be able to translate that into what the corresponding RNA mutation is and guesses at what the different protein mutations could be. We calculated some scores related to that, and then we were able to connect that up with what we already knew about mutations and treatment options and induce a subset that we could then present to the oncologist with the entire line of evidence, and they can then choose and see if that makes sense. So all of this magic, or I like to call it magic, but this was done in Clojure. And so when we first chose this, we did it because this particular working with this data, it really made sense to do it in functional style. Because the data isn't what was important, but how we worked with it was what was the crux of the system. But the more we used it, it was the perfect choice. We couldn't have done it in anything else, I think. And the thing that we particularly used is macros. So we wrote a ton of macros, and it made our life so easy. And I worked on this for almost a year, and I think the production code was about 300 lines. So it's pretty awesome. So finally was actually our front end. So we had to displace something. We really tried to do user-centric design here. So we had Clarity, and then we had Clarity's users. And we tried to engage with them throughout to try to see exactly what kinds of information mattered, to the oncologist as well as to the patient, and what was important in the conversation. We started out doing like fancy things like the physics engine. We went to town with D3, we did processing, we did Canvas, SVG, and then threw everything away because nobody liked it. What they wanted was a table, and they just wanted columns and clearly bold letters explaining important points. That's all. So that was great. We learned a lot, and we got to play with some tools. And we also tried to entice them and be like, we actually even built like a JavaScript console visualization, and we're like, you can be a power user, and it was a fail. So great. Again, just the end-to-end flow of our system is this, like I'd mentioned before. JRuby was very helpful in helping us unify, combine a lot of data, convert them into Clojure DSLs. We then expanded that and put it into the Neo4j graph. We deduced and built evidence, all in Clojure. And finally, our front end, the main technologies was Composure, CoffeeScript. We used Haml and SAS. Another, just a shout out, was from the first week we started developing, we also focused a lot on being able to go to production, because we realized no matter how little or how much we find, the use is only if Clarity and then the patient can use it. And we coded our infrastructure code in Python, and we used Amazon Web Services and Puppet to help provision things and all that. So a summary of what we did get to achieve. So today, Clarity has scaled treatment. We released production late last year. And actually, all this effort to get things working for Clarity took us about 3 months. and we were very happy with that. So they were treating about 10 patients a month, and I think they've grown. And so at that point, they had done 300 in total, and now they're close to almost increasing that by 1,000 more. So we're glad we were able to help. And we also were able to build an evidence-based system. So not only did we just give treatment options, we provided that entire history so Clearity and the patient could have a good conversation, and it was more contextualized. And then we also came up with a few alternate therapies that even wasn't there in Clarity's initial sort of guesses. So yeah, that was pretty exciting, and we hoped it was useful. So the learnings, we had some big issues, and this was a learning actually. I wasn't kidding about a lot of data. It was a lot, and it was easy to get lost. And the other part was a lot of it is very poor quality. So different universities, different researchers, everybody has a different reason why they're looking at this data and how they input it. Most of them are just open text fields where you can write whatever you want. So people will define things completely differently, and we had to really sort of think about how we would normalize this and actually know that this X and that Y actually means the same thing. That initially was very disheartening, or not disheartening, but it scared us, but we learned to live with it. People are also used to manual intervention a lot. So research scientists, all the gold is in their minds, and they like to do things manually. They like to cross-check. So getting them to trust the automation and trust code was quite difficult. As well as a lot of the tools we had to use in order to download as well as get some of this RNA and other type of data, we had to use tool sets written again by research institutions and academics. So they don't write code for production. They write code for a reason, and then they just do that, and that's great. But it's very hard to reuse. It's hard for them to maintain, and there's so much of recreating the wheel. So again, big research institution does the same thing. They all do it differently, and that's weird. So things we did to mitigate some of this was, again, like I'd mentioned, we took it from everywhere. So the common practice is normally to pick your best source. We just took it all. And we were able to do this again because when we started, we built a pipeline where after that parsing and normalizing, we weren't dependent on the source anymore. Everything was very generic. So that was the only place where we had to tweak things when the source changed. Instead of building an end-to-end that was dependent on the way the data was defined in that particular source. Another thing we started to build into our system is feedback and learning. We did this actually by just putting in code by hand right now. So when we learned some particular output of the system meant more than what we knew about it, we actually put it into our code as facts, actually. We just said, hey, know about this. That went into our Clojure realm, but our goal was to actually put it end-to-end. So when someone clicked in the front end, we would teach our system that, hey, know this additional piece of information. So some other things I just wanted to mention was that it's important, it was very important when working with this to keep up with the domain. So it's ever-changing, ever-growing. It's also very complex. So we had to learn a lot about genomics, a lot about what it meant, a lot about cancer. We had to read papers, and it was very interesting, but it was important to constantly remind, even as developers, you needed to know the domain. And then working with the research community, like I pointed out, again, it is very different than working in the industry, but this collaboration, we can see how important it is. So it was, again, something we learned, but it was definitely something that's not easy in the beginning, but you learn to work with them. Finally, legal implications as well. There's huge legal implications to working with patient data. Working with, in general, you're working with life and death. You need to be very careful. Even with the downloads and everything else, just a heads up, we did a lot of research into licenses and in what domain did the data exist, and if we can use it and why, and we had to actually apply to a lot of places to get access to things and stuff like that. As well as we encountered weird and maybe wrong data. A part of it is that this field is changing so fast that things get out of date very quickly. So something that was valid finding 6 months ago isn't anymore. So you have to keep up with the latest publications and things like that. But it turns out publications have typos. So things will be wrong even there. We found a few and then had to go and confirm with Clarity and be like, what's going on? And it was actually wrong. This was, again, really scary. It's out there. But I think oncologists and other, the people involved in actually working with the patients are aware of this, and they have a way to mitigate it, I hope. And also, finally, we touch the genome realm. There's plenty of omes that all work together to make us who we are and to affect the prognosis of this disease. So there's so much opportunity. It's very exciting. It's very interesting. So I just wanted to point that out. Finally, yeah, I just like to conclude. So I hope, we were very excited that we got something in production and it was useful and helpful, but there's still a long, long way to go. We touched on ovarian cancer. We also only touched on the genomic relevance to treatment options. There's so much to be done, and I really encourage everybody as a developer to put on your thinking hats and get involved in this community. And hopefully software can help make this progress even faster. Thank you. Yep.
Did you run it on previous patients? Did you save the model, like the software you run?
Yes, we did. So we had about 300 from Clarity, so we were able to confirm, did it match up with things that came up for them? We did. So yes, of course we did, which was good. We learned a lot from that, and that sort of was the start of the facts and kind of understanding those things. But we also, what was I going to say? Sorry, I'm blanking out. We also found that we were coming up with more options. So, yeah.
Yeah, you said you developed data. What's the size of your genome database storage size today?
I don't know the exact number. James, do you? Like, I know the number of nodes. We have over like 1 million nodes and relationships and things like that. In terms of just the size, I don't actually know.
Well, I'm thinking like if it's half a terabyte per patient.
So we don't save that straight into the database at all. So we yeah. Yep.
So you talked a lot about the size of the data on the genome side. How big was the treatment option data set? How many treatment options?
Very, very, very much smaller. So, like, do you want numbers or?
Just like order of magnitude, are there thousands? Are there?
The treatment options, hundreds or yeah, hundreds. Not at all. So yeah, that's why I didn't stress too much on it. But that's the sad part. Like those connections don't really exist in a format that was easily consumable, at least by us at that point.
Right. And then, so when you're matching, so you do all this analysis and you come up with, a description of the patient. When you're matching to that, I imagine you're getting a bunch of different sources. Is there one that stood out as very important? Like this is the most important matching is statistical data that we've got from studies, or is it kind of expert system stuff that you've got from doctors? What was the matching between, that kind of led you to a specific treatment? Does that make sense?
No, not. I don't think I fully understand.
What the connection is between a genome and a treatment?
Right. Okay. So the matching itself is that normally when they say this treatment option is possible, it'll be like blah protein is expressed or something like that. And what happens, say you have only DNA data available, there's a way to make that correlation to see if that blah protein gets expressed. And however, though, the more levels you traverse, the probability or the chances you're not really sure. So what we actually tried to do, so Clarity has their own specific test they run. So what we can do is give them that recommendation, like run this test to see if that is actually getting expressed. Then you match it up, and then that's a viable treatment option. But they had very specific things that were tried and tested. So they knew for sure that if this thing happened at the DNA level, it really meant that at a protein level, which meant that treatment was valuable. So in terms of the most important data, though, it is what's in the mind of a genomic oncologist, if you will. So Clarity actually had a lot more useful information than the oncologists themselves, because they look at it more from standard of care and just from the outward biology. And at the cellular level, people like who were working at Clearity had the most useful information. Yep.
Do you guys have open source projects that you're using in your solution? And how can like developers like us contribute to that? And then how open is like that data feeds that you got, the half terabyte? Can anybody just like?
Yeah, that you can absolutely do. So one source is the Cancer Genome Atlas data. They have all types of cancers and various types of data. So DNA, there's even a more lower versions of that, which is called like BAM files and things like that. So they have things at a very low level, as well as more, they have protein level data and all of that, all available for download. But I think they don't let it be used for commercial purposes. We'd have to read about that, but so that is a place to start to actually try and test different sort of ideas and things like that. In our project, I don't think we used anything open source. We definitely used some open source tools in order to understand the data, as well as start extracting some. So for example, from NCBI, which is National Center of Bioinformatics, we wanted to download certain subsets of genes, and they actually have developers who write some code to help you actually pass in some sort of query and get only whatever you want back. So that was, again, I think he wrote his in Python, and you actually had to get the code, use that in order to bring down that data. So that's sort of the extent to which we used the open source tools available. But there's definitely plenty, and I also think some of them are very open to developers going in and actually suggesting like refactorings and maintainability suggestions. And I can give you more examples. Like, I don't know off the top of my head, but I've definitely heard it being talked about. Cool. I think we're 2 minutes over time, so thank you very much.